All right, we are continuing talking about muscle fiber contraction and relaxation in this video. So we got started with contraction of a muscle fiber in the previous video with the excitation-contraction coupling. Again, just a little refresher, we get that action potential arriving at the neuromuscular junction, releasing acetylcholine into the synaptic cleft of that neuromuscular junction. The acetylcholine is going to bind to those acetylcholine receptors on the membrane of the muscle cells, and that is going to trigger those sodium ion channels to open, transferring that action potential into the membrane, that sarcolemma, and causing it to travel along the T-tubules, which are going to release calcium ions, and that is where we are going to pick up in this video. So these calcium ions are really, really important for muscle contraction because they are responsible for the movement of the troponin and tropomyosin proteins off of the actin binding sites for the myosin heads. All right, and so what actually ends up happening with contraction is the actin and myosin filaments are going to slide past one another to shorten that muscle. And then the reverse is going to happen when all of that calcium either gets used or resorbed, so taken back up and removed from that cell, so getting it back out of that sarcoplasmic reticulum, getting it out of that sarcoplasm, so that it is not moving those regulatory proteins anymore, and that's going to allow the muscle to relax. So let's talk about that actual contraction part, and the contraction is done through something called the sliding filament model or sliding filament theory of muscle contraction. So when a motor neuron signals a muscle cell to contract, the thin actin filaments are going to be pulled and slide past those thick myosin filaments within the sarcomeres of the muscle fibers. That sliding can only happen when the myosin binding sites that are on the actin filaments are open. So they are no longer blocked by the troponin tropomyosin complex. And that is done through calcium ions entering that sarcoplasm. So the troponin tropomyosin complex, those are those two regulatory proteins. Those are going to normally block the heads of those myosin filaments from binding to the active sites on those actin filaments, so that our muscles are not constantly in that contracted state. That can actually lead to a lot of issues. Kind of brief rabbit trail from there. So you've probably heard of something called lockjaw or tetanus. And so tetanus is a toxin produced by a particular bacteria. That bacteria is Clostridium tetani. And it's producing that tetanus toxin, and tetanus is going to mess with this relaxation step of muscle contraction and relaxation, and it's going to prevent the muscles from actually being able to relax, and so they actually stay in their contracted state, which is what causes locking of the jaw. So that's where the name lockjaw comes from, is because the muscles in the jaw are contracted and are not relaxing. That causes the jaw to lock up. It's actually something called contractile paralysis. So the inability to move your muscles is paralysis, and there's two different types, contractile and relaxation paralysis. So contractile paralysis is what's going to happen with that tetanus toxin, among other types of toxins out there, and it's going to prevent your muscles from relaxing, and that can actually cause a lot of issues. People have actually broken teeth and fractured bones and stuff because their muscles are contracted so tightly and will not relax. You can take microbiology with me if you want to learn more about tetanus toxins. So getting back to our sliding filament model, so troponin, tropomyosin complex, those regulatory proteins, are normally going to prevent those myosin heads from binding, so that our muscles are not constantly in their contracted state. And so to start this process of contraction is to get those calcium ions in and bound to the troponin molecule. which is going to allow the tropomyosin protein to slide away and uncover those binding sites on actin, which is going to allow the myosin heads to actually bind onto those binding sites and form something called cross bridges. And those cross bridges are that connection of the myosin heads to the actin filaments. And then those myosin heads are going to pull those thin actin filaments, sliding them past the thick myosin filaments and closer to the center of that sarcomere. And so those filaments themselves are not changing in length, they are just sliding past one another. Each of those myosin heads can only pull for a very short distance before it reaches its limit, and it has to be reset before it can actually pull again, so that step's going to require ATP. And so this can change a little bit how the muscle contracts, how long it's able to stay contracted, and stuff like that. So this is, again, our sarcomere model here. So we've got the thin actin filaments here in green with the troponin and tropomyosin proteins on them. We've got our thick myosin filaments here with the little myosin heads. And so what we're doing in this sliding filament model is ending up pulling these Z discs formed by the connection of actin filaments closer to that M line at the middle. All right, so we're shortening this H band quite a bit, pulling those Z lines closer to that M line in the middle. But again, the actual size of the filaments is not changing. So actin and myosin filaments are not getting shorter or longer. They're just sliding past one another. And I've got a little animation here in a couple slides to show you. So ATP, like I said, is required for that cross-bridge cycle to keep going, especially if we need that muscle contraction to be sustained and keep going and not relax too soon, because obviously there's some cases where we don't want our muscles to relax immediately after contracting. So we're going to have that contraction happen. So calcium's going to come in and bind to troponin. And that troponin anchors the tropomyosin filaments here in place, blocking those binding sites. So calcium comes in and binds to troponin, and then troponin is going to now allow those tropomyosin covers to move, exposing those binding sites. And then the myosin heads are going to bind in there and then pull that actin filament in one particular direction, and that's going to force some of those calcium ions to release. All right, and then we come in and have that ATP, that energy source, coming in to help continue this. So this is required for, again, that repeated contraction, so needing to keep contracting those muscles or keep them contracted, we have to have that energy. So you can think of these myosin heads as kind of like paddles or oars that you would use for like a kayak or a canoe or something like that. And they are paddling the actin filaments, if you want to think of it that way. That may or may not make sense to you. It's totally fine if it doesn't. But they're kind of paddling those actin filaments, getting them to move in one particular direction. And that obviously requires energy. So what we need to do is have that ATP come in and help out with that repetitive pulling. And so what you're seeing here is initially we've got ADP and then this P sub I molecule. So ADP is adenosine diphosphate, and then the P sub I represents a single phosphate molecule, and then ATP is adenosine triphosphate. So this ADP has 2 phosphate molecules, ATP has 3 phosphate molecules. And so to get from ADP to ATP, we have to connect this extra phosphate into the ADP molecule, and that's going to give us that ATP, and then using that ATP is going to break off one of those phosphate molecules, giving us ADP again. And so that can kind of keep going in a not infinite cycle, but it can keep going for quite a while as long as it has all of the resources necessary and it's still getting the signal from the neurons. All right, so that myosin head attaches to actin. Well, we have that ADP form, plus that inorganic phosphate molecule that are bound to that myosin head. So they're attached to that little myosin head or paddle. And once that phosphate is released, it causes myosin to form a stronger attachment into those actin binding sites, really holding on to them very, very tightly, helping to pull them towards that M line. And then this is going to move these filaments about 10 nanometers, which is a very, very small distance. It's a thousandth of a meter. Really, really, really tiny distance, which is why we need to keep rowing, essentially. So we need to keep paddling with these myosin heads in order to get full muscle contraction, for these bigger muscles especially. So we're going to pull towards that M line a little bit. This is called a power stroke movement. And then it reaches that 10 nanometers approximately. This movement is called a power stroke. And so without ATP, the myosin head is not going to detach from actin. So unless we have that ATP coming in, that myosin head is going to stay bound to actin until either we get ATP bound to it or the troponin-tropomyosin complex kicks it off, as their job as regulatory proteins needs them to. All right, so we've got the ADP binding site and then ATP binding site on the myosin heads, so slightly different binding sites. So once we pull that actin filament, it's going to kick off that ADP and phosphate. Those are going to go get recycled elsewhere in the cell to create ATP in the cellular respiration process, which we are not getting into in this class. That is a very complex process that we do not need to know for anatomy. And then we have that ATP coming in, being signaled to come in and attach to the myosin heads through various signaling pathways that, the cell is sending out saying, hey, we need to keep contracting, we need this ATP to come in, and that's going to allow that myosin head to detach from the actin binding site, and then that is going to reset that myosin head, breaking off that phosphate molecule, transforming that ATP back into ADP, and then it is going to come back and rebind to those actin binding sites and pull those actin filaments another roughly 10 nanometers. And so that's going to keep going as long as there is enough ATP available and there is still enough calcium available to keep that troponin-tropomyosin complex from blocking those binding sites. So here's that animation I was talking about. So we get those calcium ions come in, pull that troponin-tropomyosin complex out of the way, allowing those myosin heads to then bind to actin and pulling them. All right. So again, sliding filament model. So those actin and myosin filaments are just sliding past one another. They are not shortening themselves. And we will continue talking about types of muscle fibers in the next video.